For this next set of notes, you need to have your cut out pieces of, well, shapes. They're cut out pieces of paper. And I'm going to apologize in advance. You are not going to enjoy this, but it is really important that you understand how DNA is put together. And so we are going to go through that step by step. And I am going to start with the shape that looks a little bit like a house, a shape that looks a little bit like a plus sign, and these are going to make up the sugar phosphate backbone. So one of these is a sugar, one of these is a phosphate. This is the phosphate. So I'm going to label this with a P, O, double bonded to an O, bonded to an O, and then there's an O on this side with a negative charge. I'm going to put that right there for now. I'm going to now label my sugar. So my sugar looks a little bit like a house, where I've got an oxygen bonded to a carbon, 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 and then I have another carbon up here. And these things stick together to make my sugar phosphate backbone. So now I'm going to use my glue stick and I'm going to start and I'm going to glue this in. And then there is actually going to be a bond that forms right here between my sugar and my phosphate. All right, grab another one of those. We're going to continue to make the backbone. So I've got my P double bonded to O. And if I glue this in, it attaches to this sugar. So I've put this here. This is carbon number 1, carbon number 2, carbon number 3. And the phosphate is going to connect to the sugar at the 3' carbon. So this is carbon number 3. We call that the 3' carbon. Now this phosphate is going to also be attached to a sugar. So I'm going to need to have another sugar here. Go ahead and label this. And then I'm going to glue this in as well. And then I'm going to bond these together because this phosphate is attached to the 5' carbon, carbon number 1, 2, 3, 4, 5. So this is the 5' carbon. We currently have 2 sugar phosphates, 1 and 2, and we know that a nucleotide is made up of a sugar, a phosphate, and a nitrogenous base. So I'm going to start with this shape right here, and I'm going to glue this in where it is connected to the carbon. We have now 1 nucleotide, and we're going to make another nucleotide right here. So I'm going to need to pick another one. I'm going to pick this one right here. And now I have a nucleotide here and a nucleotide right here. Now this is just one side of my DNA. My DNA is double-stranded, so I'm going to need another strand of DNA here. They're connected through the bases. So let's look at which base would attach to this one. And it turns out it would be this one here. So I am going to glue that one in. And I'm putting a little bit of space between them so that I can draw some hydrogen bonds. So let's put 2 hydrogen bonds between these. The nitrogenous base is connected to carbon number 1, which means that when I connect it to this sugar, I'm going to have to connect it at carbon number 1. So first start by labeling the parts of your sugar. With the parts of my sugar labeled, I know that this must be carbon number 1. So in order to bond that to my nitrogenous base, I'm actually going to need to flip this over. So I'm going to glue this in, but I'm going to glue it in flipped over. And then I'm going to make a bond between carbon number 1 and that nitrogenous base. But we know that a nucleotide is made up of a sugar and a phosphate and a nitrogenous base. I currently have my base and I have my sugar. I now need to add my phosphate. So again, we are going to start by labeling our phosphate. And this phosphate is going to get glued in like this. And the sugar and the phosphate are connected to each other. The phosphate is connected at the 5' carbon. So again, if I start at the oxygen and I count clockwise, 1, 2, 3, 4, 5, this is carbon number 5, that's the 5' carbon. So I now have a whole nucleotide right here, and I have a whole nucleotide right here. Let's go ahead and build the partner to this one. That base is going to pair up with this base. So let's go ahead and glue that in. And because these two shapes have 2 hydrogen bonds, I'm going to make sure I have 3 hydrogen bonds between my other set of shapes. And these shapes represent the nitrogenous base. Again, the nitrogenous base is connected to a sugar, so let's start by labeling our sugar. And the nitrogenous base is always connected at the carbon number 1. So go ahead and connect to the carbon number 1. And then this sugar here is also connected to a phosphate. Label your phosphate, and then attach that phosphate at carbon number 5. So now I've got a whole nucleotide here, and I have a whole nucleotide here. And those nucleotides have to be connected to each other. So go ahead and add a bond between your phosphate and the 3' carbon of the other nucleotide. Let's go ahead and label our nitrogenous bases. So if this is a large base that has 2 hydrogen bonds, then I know that it must be an A. And A is always bonded to T. I'm writing it upside down for a reason. And then if this is a large A, this one's also large. It has a different shape and it has 3 hydrogen bonds, so this must be a G. And then G is always bonded to C. So again, I know that I wrote that upside down. And the reason that I wrote it upside down is because you should be able to look at your DNA from either side and be able to read it in a specific direction. And we will talk about that specific direction in a little bit. From here, I'm going to keep building my DNA one piece at a time. So this carbon, I know, is connected to a phosphate. And this phosphate is connected to a carbon that makes up part of the sugar. And this sugar on carbon number 1 is attached to a base. So I'm just going to pick a random base. I've picked this one, so I'm going to glue this one in. Okay, if I flip this over, I can also add another nucleotide here to pair up with this one. So I'm actually going to put the nitrogenous base in first. I don't know how this is all going to work, but we're going to try it. So I've added my base, and I'm going to add my sugar and my phosphate. So this time, this phosphate is bonded to carbon number 3. And carbon number 1 is bonded to the base. And this pair of bases is a C with 3 hydrogen bonds to my G. But this is not done, because this is the sugar. It needs to be bonded to a phosphate. And then I can do this all over again. So I flipped it back over. I'm going to add another whole nucleotide. And I'm going to flip it over so that I can build the other side. And this is really important. This is going to be the last one I draw, which means that this phosphate here actually has an OH. I'm also going to add some additional details. So down here, this carbon also has an OH. Because it isn't bonded to anything else, it has an OH right there. If I flip this over, this phosphate on top really needs to have an OH because it hasn't been bonded to anything else. And then this carbon at the bottom also needs to have that OH. And those OHs are important because the OHs are where things can bond to. So if I were to add another nucleotide to this, I would need to remove this OH here and attach a phosphate. Now, if we look at what a nucleotide looks like, we can see that there's a phosphate that looks like this, and it's got this OH at the top. Now, we know that when we do dehydration synthesis, you remove water. So what we're going to do is we're going to remove this OH, we're going to remove that H from the phosphate, and that's going to leave me with the ability to have a C bonded to the O from the phosphate and then the P. So it's going to end up looking like this. The C with the O from the phosphate and then the P from that phosphate. And dehydration synthesis is not only how you put together a nucleotide by removing an OH and an H in order to make this bond here, removing an OH and an H in order to make this bond here, but it's also how you connect one nucleotide to the next nucleotide because you've removed an OH from an H in order to make this bond right here. You'll notice that when we look at sugar, you can see that it has 3 OHs. And each of those OHs is going to be used to bond a different thing. You are going to bond the nitrogenous base to carbon number 1. You're going to bond the phosphate to carbon number 5. And then you bind one nucleotide to another using carbon number 3. So let's update this with a labeling of one of those nucleotides. So a nucleotide consists of a sugar, a phosphate, and the nitrogenous base. And what's really important to realize is that the phosphate that's included in the nucleotide is actually the phosphate that is connected to the 5' carbon. So this thing that I have circled here or boxed, this is a nucleotide. And then DNA has directionality. We can read DNA in a specific way, and you have to know which direction the strands of DNA are actually going in. The way that we label these, the way that we identify this, is we say that there is a 5' end and a 3' end of the DNA strand. And so the way that we do it is we look for where is the 5' and where is the 3' carbon. So I'm going to look at this nucleotide right here. So I've got my phosphate, my sugar, and my base, and that's going to make up one nucleotide. I'm going to start at the oxygen, I'm going to count my carbons around. 1, 2, 3, 4, 5. It does not matter which sugar I'm looking at. The 5' carbon is always going to point towards the 5', this is how we write it, the 5' end of the DNA. The 3' carbon is always going to point towards the 3' end. We write it 3 little apostrophe, end of the DNA. Another way to remember this is that the oxygen always points towards the 5' end. So if I'm looking at a strand of DNA like this, my oxygens are pointing in this direction. That is the 5' end. Looking at my other strand of DNA, if I count my carbons around, so I'm going to start here, count clockwise. This is carbon number 1, 2, 3, 4, 5. The 5' carbon points towards the 5' end. And the 3' carbon points towards the 3' end. And remember, my oxygens, they're now pointing down. My oxygens are always pointing towards 5. So what you should notice here is that my DNA strands are running in opposite directions. We've got a strand that's running from 3 to 5 this way, and then our other strand is running from 3 to 5 this way. That's going to be really important later. For right now, you should be able to identify a nucleotide by looking at the sugar, the phosphate, and the base, and knowing which phosphate it is that makes up that nucleotide. If you're asked this question and you circle this base, this sugar, and this phosphate, that would actually be incorrect. That is not a nucleotide. A nucleotide has the phosphate connected to the 5' carbon. You should also be able to identify the 5' and the 3' ends of the DNA. You should be able to tell me which nitrogenous base I have based on whether or not it is a large base, a small base, and the number of hydrogen bonds that there are. You should be able to tell me that DNA is double-stranded and that the backbone is made up of these alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphates. It would keep going. That is the backbone. And the two strands of DNA are held together by the hydrogen bonds in the middle. Let's just go ahead and label that. And that's all.